Good morning or good afternoon, everyone, and welcome to Genome Webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Single-Cell Sequencing Reveals Clonal Heterogeneity in Acute Lymphoblastic Leukemia. This webinar is sponsored by Mission Bio. Our speakers today are Dr. Jan Kool's, Professor of Genetics at the Catholic University of Leuven in Belgium and a group leader at VIB-KU Leuven, and Dr. Lucia Alberti Cervera, a postdoctoral researcher at VIB-KU Leuven. You may type in a question at any time during the webinar, and you can do this through the Q&A panel, which appears on the right side of the webinar presentation. We will ask our speakers your questions after the presentations have concluded. Also, if you look at the bottom tray of your window, you will find a series of widgets that can enhance your webinar experience. So with that, let me turn it over to Dr. Kool. Please go ahead.
Thank you very much. So let me first give a brief introduction on acute lymphoblastic leukemia, which is the disease that we will be studying today in this webinar. And then afterwards, Lucia will come back with details on the experiments. So acute lymphoblastic leukemia is a leukemia that is most common in children. It has a peak incidence at 2 to 5 years of age. It is also occurring in adults, but is much more rare in adult cases. Overall, if we take all the adult and the pediatric cases together, it's about 17 cases per million people per year that we see as new diagnosis of this disease. ALL can be subdivided in two subtypes. The most common subtype is the B-cell variant, where the B cells are affected. That's 85% of the cases, and 15% of the cases are of T-cell origin. Treatment of acute lymphoblastic leukemia is a very intensive multi-agent chemotherapy treatment that lasts for about 2 years, consists of many different drugs that are used, and it's a long process, especially if you know that this is mainly young children that need to be treated. Luckily, this 5-year overall survival for the children is very good. It's more than 90%. In adults, however, this is still much lower and is about 50 to 60% with the current treatment protocols. The major problem that we see in acute lymphoblastic leukemia is relapse. So once most of the patients respond initially to therapy, they respond very well, but unfortunately some patients relapse where the disease is recurring, and this is the 10 to 50% of the patients in the children or adults that do poorly then on therapy. Once a patient relapses, the prognosis is rather poor. When we look at the origin of B-cell or T-cell ALL, we need to look at the genetics and so look at the mutations that are present in this disease. And in B-cell ALL, we see a lot of transcription factors that are often inactivated by deletions or mutations or chromosomal rearrangements. And in addition to that, we also have a lot of signaling mutations in kinases or other signaling proteins such as RAS or signaling receptors. In addition to that, there's chromatin modifiers that are often mutated. P53 can often be mutated in B-cell ALL, and also other cell cycle genes are often mutated. You see, you have more or less the same picture in T-cell ALL. We have their overexpression of transcription factors, typically TLX1 or TLX3. HOXA factors can be overexpressed and block the differentiation of the T cells. And in addition to that, we have a lot of mutations, again, in signaling proteins, chromatin modifiers, other signaling proteins. And one particular mutation that is very specific for T-cell ALL is the Notch pathway that is very often mutated. Almost all T-cell ALL patients have mutations in either Notch1 or FBXW7. And so that's a very specific and important mutation in this disease. These mutations occur all together. And so what we now understand more or less is that the several years during which acute lymphoblastic leukemia is developing in a patient, it occurs through a stepwise accumulation of different mutations. And at diagnosis of a patient, we typically see 5 to 10 different mutations that are present in the major leukemia clone. Now, since this is a stepwise mutation, as shown on this slide here, mutation 1, then some cells acquire a second mutation, mutation 2, some of these cells will acquire a third mutation, and so on. This is not a linear process necessarily and can be a kind of a branched evolution process. So you have the green cells, which have already 3 mutations. Then some of those cells will acquire a 4th mutation. These are the yellow cells. But other cells, other of those green cells, can acquire another 4th mutation, which is then shown here as mutation 5. This is then the purple cells. So you could have a diagnosis, different subclones that are present in a particular patient. And that's something that we try to understand now with this single-cell sequencing. So the main research questions that my lab is trying to address is in what order are these mutations acquired? If we know that patients at diagnosis have 5 to 10 different mutations, is there a specific order through which these mutations are acquired, or is this a random process? In what cell type are the mutations acquired? Is this in stem cells or in lymphoid progenitor cells? How many different clones are present at diagnosis? Is this different from patient to patient, or do we always see the presence of some subclones? And finally, how do these different clones evolve during treatment is also a 4th question that we try to address. And so for these questions, it's definitely needed that we use single-cell sequencing analysis, because if we look at bulk sequencing, we can get an idea of the different subclones present, but it's always difficult to determine whether different mutations are present in the same cell or in different cells. If we look at single-cell sequence data, we can now try to see whether these mutations are in the same cell or in different cells. And so with this, I hand now over to Lucia, who will get you through all the details of this single-cell sequencing.
So thank you, Jan, and hello to everyone. So after this very nice introduction on ALL, I will focus now on my postdoctoral research project. So in my project, I applied the single-cell sequencing with 2 main objectives. The first one is to investigate the cellular and the subclonal composition of acute lymphoblastic leukemia at diagnosis. Like this, we expect to get insight about the level of heterogeneity both at the DNA and the RNA molecular levels. And this is before treatment. So a second aim of the project is to follow the cellular and the subclonal evolution in ALL during the treatment. Like this, we can get insight about which are the clones that are more sensitive to the treatment and disappear, which ones are more resistant and get expanded, and also monitor the appearance of new clones. Eventually, we can get insight about the detection of early clones at diagnosis or during early treatment that might confer a high risk for treatment failure or a relapse. Now, to investigate all this, this is the research methodology that we follow. The first step, which is very important and is crucial, is the collection of patient samples and the cryopreservation of them. We have a very nice collaboration with both the hematology and oncology departments of University Hospital Leuven, and upon the diagnosis of a new ALL case, either adult or pediatric, we receive a bone marrow and blood sample before the treatment. And then, following with the clinical follow-up of the patient, we collect additional samples. These include 2 blood samples during early treatment, but also a blood and bone marrow at day 35, which coincides with the clinical assessment of minimal residual disease. And then, if the patient, in case there is a relapse, we reactivate the collection of samples. All the samples are very fresh. Within an hour, we have them in the lab and start the processing. For that, we isolate the mononuclear cells by gradient centrifugation with Ficol and cryopreserve the cells. And then we prioritize for the single-cell sequencing TLL cases, which is an aggressive subgroup of ALL, but also VLL cases that had a high white blood cell count at diagnosis or that were MRD positive at day 35. Then from each vial, and in parallel, we process few thousand cells with the 10x genomic platform for whole transcriptome RNA sequencing, and few thousand cells with the with the Tapestri platform from Mission Bio for targeted single-cell DNA sequencing. So like this, we can have from each patient and time point the cellular composition of the leukemia, so the identity of the cells based on the gene expression profile, but also the clonal composition of the leukemia based on their mutational landscape, so how the mutations co-occur at each individual cell. Then there is an additional platform at the Institute, GNTsec, that enables RNA and DNA sequencing from the stem cell, and which we'll also use for a few samples. But the focus of today's seminar is the single-cell DNA sequencing with Mission Bio Tapestri Platform. So first of all, I would like to introduce you this platform. Then I will briefly show you the custom panel that we generated for this targeted sequencing. And finally, I would like to show you some interesting data on the clonal architecture of the first patients that we have sequenced. So let me start with the Tapestri platform. Tapestri, it's a commercially available platform from Mission Bio and is for high-throughput genomic analysis at the DNA and the single-cell level. It's a targeted approach, meaning that you have to select which regions of the genome you want to sequence. And for that, the company already provides 4 commercial panels on the catalog, but also the option to design a custom panel by selecting your own genes or regions of interest. So they have a more general panel that includes genes that are highly mutated in several solid tumors, and then 3 other more specific panels, either for acute myeloid leukemia, chronic lymphoblastic leukemia, or myeloid malignancies. Each of the panels, the multiplex level is about 300 regions, and the cell throughput is between 5 and 10,000 cells. This is really sample dependent and depends on the cell type. You will see later that for leukemia, we are in the lower part of this range. And then this technology, it's for detecting single nucleotide variants, but also small insertions and deletions. They are also working on inferring copy number variation data, information from this data, but it's not for detecting fusion genes or rearrangements. This cannot be done with this targeted approach. The major insight that we can get with this technology is that if you do a bulk sequencing, you can check, for example, you can see that, for example, in a sample, 40% of the cells will have a blue mutation and 40% the red mutation. But by going really at the single cell level, you can see how these mutations co-occur and identify that 20% of the cells in the sample will have both the blue and the red mutation. And then another advantage is that you can really detect rare SAC clones below 1% that otherwise, or most of the times, are missed in bulk approaches. All right, so now I will briefly show you the workflow and how the platform works. First of all, it's the sample preparation to make a single cell suspension that is loaded into the Tapestri platform. The loading, it's about 100,000 cells in a concentration of 3,000 cells per microliter, and then it takes place the cell encapsulation and the barcoding into 2 separate steps. So first of all, the technology is a macrofluidics, and the cells pass through oil emulsion and are encapsulated into droplets together with lysis buffer and proteases. The cells are lysed, and the proteases digest genomic DNA so that you have access to digest the DNA binding protein so that you have access to the genomic DNA. And after that, the lysate has to pass again through the platform and is merged with droplets containing barcoding bits. And each of these bits is coupled to oligonucleotides that contain the handle for the library, the cellular index, and also the forward primer for the gene of interest, so that each of the cells will receive a combination of the about 300 primers to amplify the targeted regions. After that, and now that the cells are labeled, they can be pulled together and the library preparation and sequencing can be performed. All this process up to the targeted amplification can be performed in 1 day, and although Tapestri only have 1 encapsulation site, it's very feasible to process 2 samples per day, and then the library preparation in a second day with 8 samples in parallel. So at the end of the week, it's very feasible to process 8 samples, having 1 instrument and 1 person running the experiments. Then after the sequencing, for the data analysis part, Tapestri also provides 2 softwares. The first one, which is for the processing of the raw data, integrates with a high-performance genomic platform, Bluebeam. And what it does is all this, the timing of the reads, alignment, cell calling, and genotype calling. And it provides several output files, including quality metrics, a cell report, and the most important file, which is all the list of annotated variants. And then these variants can be filtered in a second software, which is the Tapestri Insight. And this is also a very useful software for reviewing the variants and also for a first exploratory analysis of the clonal architecture of the sample. Then you can export all the data, and further and more complex analysis can be done in other platforms like R or, for example, MATLAB. Okay, so now I'd like to move to the second part, which is the panel that we generated. So as you have heard at the beginning, our focus or research focus is on acute lymphoblastic leukemia, and none of the commercial or available panels was on ALL. So we decided to design our own panel to cover the mutational hotspots of ALL. For that, the design approach that we followed, it's a focus approach, meaning that instead of selecting full genes, we really selected specific regions that are reported as the most mutated in ALL. For that, we were mainly based on a PECAN database. This is a pediatric cancer database that includes all the reported mutations today for pediatric cancer. It also includes a very good visualization tool. So, for example, I show you here for Notch1 gene, which is a hallmark for T-cell commitment and also very important in T-cell development. And as Jan mentioned at the beginning, is mutated in most of the TLL cases. You can see here what are all the mutational hotspots for pediatric TLL, and we could really zoom in and select those genomic coordinates that are relevant for our panel. We did this for all the genes, and also included variants that are reported in the COSMIC database that is mostly for adult cases, and also some variants that we identify in our lab with previously sequenced patients. So as a result, this panel covers 305 genomic regions of on average 200 bases amplicon size, include 110 different genes, and spans more than 1,300 reported mutations. It's mostly single nucleotide variants, but also covers some insertions and deletions. So what are those genes? Here you have a nice overview of the 110 genes that are included in the panel. They are ordered alphabetically, and the size of the text, it's proportional to the number of amplicons that cover the gene. The color is proportional to the number of mutations that are reported for that gene. And you can see that, for example, for some genes like Notch1, there is a good correlation with a very high number of amplicons covering the gene and very high number of reported mutations. However, for other genes, it can be like P10, for example, that most of the mutations are located in few spots. And for other genes like SMARCA4, that the mutational pattern is more spread over the gene. All right, so we had the chance already to run and sequence 24 samples with this panel. And the first thing I would like to show you is the performance of the panel. So now in this plot, what you can see is that each bar is one of the 305 amplicons, and what is plotted in the y-axis is the average number of reads that on average a cell received per amplicon. So you can see that the sequencing is not equally distributed across all the amplicons. We always have some very high performing amplicons on the left and lower performing amplicons on the right. But this is not dependent on the panel, depends on the selection of the genes. This is really dependent on the panel and the selection of the genes, but not on the sample. So this plot is the average of 24 samples, and for all the samples, the redistribution was very similar. Always the high performing and low performing amplicons. These 24 samples are from 9 different patients and include both BLL and TLL leukemias from either pediatric or adult cases. We also sequenced 2 xenograft samples and 1 ALL cell line. And the panel performance was always very similar. So this panel, it's validated and can be used for all these kind of samples. Here you can see what are the high and low performing amplicons. It can be that in the same gene, like for example, JAK1, one region amplifies very well and other region is less amplified. But overall, to be able to annotate a variant with confidence, we need at least 10 reads in that region. And you can see that there are 15 amplicons that are below the threshold. But overall, we expect that from each of the cells, more than 95% of the amplicons will have sufficient coverage to annotate mutations, which is pretty good. Then the next thing I want to show you is some metrics on these 24 samples. So now each of the the dots, each one of the samples, and the X labels is just the patient code. And first of all, you can see the number of cells that we were able to obtain after passing quality filters for each of the samples. On average, we obtain about 4,000 cells. A maximum is a bit more than 6,000. And you can see that for one of the samples, we really obtain less cells, but it's because we loaded below the 3 times below the recommended concentration. So this was expected. But overall, we have already sequenced 100,000 cells that were loaded with the Tapestri platform. And then the coverage, which is the number of reads per amplicon and per cell, you can see here that was very high for all of the samples. Normally, the recommended average coverage per sample is 80, so that you can also annotate variants from the low-performing amplicons. And you can see here that for most of the samples, we are above 100. And normally you cannot get this level of coverage and resolution if you will just do, for example, a whole exome or whole genome sequencing. And then another metric to take into account is the allelic dropout rate. So allelic dropout can occur when one of the alleles of an heterozygous locus fails to amplify, and therefore is not sequenced. As a consequence, it can be that cell that is called wild type or homozygous for a variant, depending on the allele that is lost, instead of true heterozygous. So this rate was calculated for all of the samples. And you can see here that is about 5 to 10%, and this is way below the other reported single-cell technologies. So overall, we are really satisfied with the performance of the panel and also with the quality of the data. And I have to say that this panel is currently available as a pre-designed option, so that you can start the design of your panel with this set of genes and also remove or include some other genes. Mission Bio is also planning to make it commercially available very soon. So with this, I'd like to move to the last part of my talk, which is a bit more the biological part. So first of all, the first step is that we receive from each of the samples thousands of variants. So the first thing we need to do is remove those variants that have low quality and that are present in low frequency. I just want to show you here the example for few samples to focus on the thresholds that we use. So first of all, we remove variants that are genotyped with a low quality, but you can see that the majority of the genotype is assigned or called with high confidence. Then a variant to be called with confidence needs at least a coverage of 10 reads. So we remove all those genotypes that are covered by less than 10 reads. And if it's the variant phenotype, at least 20% of the reads need to be of the variant allele. So the variant allele frequency needs to be at least of 20%. Then we remove variants that are genotyped in less than 50% of the cells. And we also exclude cells that in which less than 50% of the genotypes are present, so that contain low information and are not informative for clustering. And finally, this filter is to set the sensitivity threshold. So we remove all so a variant to be selected needs to be present at least in 0.5% of the cells, in 1 every 200. And you can see that most of the variants don't pass this filter. So after applying this, normally you obtain 100 to 120 good quality variants per patient. I show you here on the left 3 TLL pediatric 3 LL patients, and on the right 1 pediatric BLL patient. And you can see that these variants can have different coding impacts, and this is about 0.5% of the variants, a bit more for the XF100 patient. Within the coding variants, the majority are missense, meaning that they imply a change in the corresponding amino acid. Now, for all these patients, we had both a bone marrow and a blood sample. So what we did next is to really compare the percentage of mutated cells for each of the variants in blood and the bone marrow. So now in this plot, each of the dots is one variant, and the diagonal will be a perfect correlation. And what I hope you can appreciate is that the frequency for each of the variants in blood and bone marrow, it's very similar. There are a few ones with the variants that are labeled, but overall, the leukemia seems to be very similar in blood and bone marrow, at least at the diagnosis. And then another observation is that the variants seem to be present either in the majority of the cells, so most likely germline variants, or they are very subclonal, so they are present in a small proportion of the cells, typically less than 10%. Interestingly enough, we still do observe some variants, like for example in the XF100 patients that are at intermediate frequency. Now, all this kind of analysis could be done with a bulk approach. So what it's really the interesting part of the single cell approach is to see how these different variants can co-occur in the same cell, and so that we can explore the clonal architecture and composition of the leukemia. So for that, and before showing you a few examples, we further selected those variants that can be the most informative and can have a major value in the evolution of the leukemia. So first of all, we filter out all those variants that are most likely a germline mutation, because we have a corresponding RNA sequencing data from each of the samples. So we know the exact composition and percentage of healthy or at least non-leukemic cells that are in the sample. So this threshold is still conservative. And then we focus on the coding variants, and from the coding variants, we exclude the synonymous ones. And then it's very important that a variant is genotyped in the majority of the cells, and we also select variants that are already referred as pathogenic or annotated as pathogenic. So with this, I would like to show you 3 examples about the clonal architecture in 3 different patients. The first one, I will start with an example at diagnosis only. So how is the leukemia at diagnosis? I'll show you here the clonal architecture based on 7 of the variants that we detected in the XF97 pediatric TLL patients. These are well-reported mutations in the JAK1, NOTCH1, USP7, DNM2, and JAK3 genes. And in the table, you can see that based on the different combinations of occurrence of these 7 variants, we can detect 7 major clones and more than 80 very rare or small clones that are present in less than 1% of the cells. So if we look at how is the distribution of these different clones, now in the lower bar you have the peripheral blood sample, and the upper bar corresponds to the bone marrow sample. And what we can see is that there is, first of all, a major clone in red. This one comprises more than 50% of the cells, both in bone marrow and the blood. And this clone contains, it's heterozygous for 5 of the variants. Then we have 3 other smaller clones that are present at a frequency of less than 10%, and they are heterozygous for 4 of the variants in different combinations. These smaller clones are also present both in the bone marrow and blood, but at slightly different frequencies. And there is 1 clone in light purple that is labeled as wild type, because the cells in this clone are wild type for these 7 variants. So this is the clone that contains the healthy cells. And you can see that it's the only one where the frequency is really different in bone marrow and blood. But this is consistent with the clinical data and also with our single cell RNA sequencing data, in which we observe that the bone marrow have a higher leukemia burden. So leukemic cells is higher in the bone marrow. Then another interesting information that we can obtain from a single cell approach is the zygosity of the cell. So we can really distinguish if a variant is either heterozygous or homozygous based on the variant allele frequency of each individual cell. So you can see that it's highlighted in light blue that we have 2 clones. The second one in blue that has a homozygous variant for NOTCH1, and this is 8% of the cells, so it's the second major clone. And then we have another one, very small, in orange that contains an homozygous variant for a DNM2 gene. This is a really valuable information and can be really interesting, but we also have to be cautious. We have to be careful interpreting this data, because as I pointed before, we are aware of the allelic dropout rates. So those clones can contain cells that are the result of allelic dropout and not truly homozygous. It's very difficult to distinguish truly homozygous from allelic dropout rate, but one thing we can do and check is at the metrics of each of the variants. So we can review the variant, and for example, we focus now on the Notch1 homozygous variant. Based on the variant allele frequency, you can see that the blue clone, it has a variant allele frequency close to 100%, so it's homozygous. And now if we compare this clone to the heterozygous one, for example, the first red one, and check at the quality of each of the cells, the genotype quality, you can see that there is a good proportion of cells in the homozygous clone that has a decreased quality compared to the heterozygous clones. And the same holds true when we look at the read depth. So the average reads per cell of that variant is also decreased, indicating that some of the cells might have lost the allele. And this also holds true for the other variant. If we look at the DNM2, we can easily spot what is the homozygous clone with a variant allele frequency close to 100. But again, some of the cells have a reduced quality and read depth. So although it's a nice feature, we have to be careful interpreting. But the Notch1 clone, which is 8%, for sure contain some of the cells that are truly homozygous. All right, so that's the first example. And what we can say from here is that indeed ALL is heterogeneous at diagnosis because we observe several subclones. In this example, for example, one major clone and some other minor clones. But now what is really interesting to see is what happened to these clones during the treatment, once the patient started treatment. So for that, I would like to show you another example with another patient. This is another pediatric TLL case, the ESF100 patient. And for this one, we not only had samples at diagnosis, bone marrow, blood, but also at 3 different time points, including minimal residual disease. So overall, 6 samples over 4 time points. And we sequenced more than 28,000 cells and detected 143 variants. Then I show you here the clonal architecture based on 6 of the variants on the Notch1, WT1, and zinc finger protein genes. And again, you can see in the table different combinations for these mutations. And if we check how is the frequency of these clones, first of all, at diagnosis, we see in red, it's the clone that contains wild-type cells, but then again, we observe a major clone in blue, and this clone contains 5, it's heterozygous for 5 of the variants. Then we also see a collection of minor clones that are present both in bone marrow and blood at similar frequencies. And interestingly, as before, we don't observe clones that are unique either in the bone marrow or in the blood. It's always the same clones. So what happens when the patient starts with the treatment? Here you can see the rest of the time points. And what we see is that the percentage of wild-type cells increases over time, and this coincides with a gradual but rapid eradication of leukemic cells thanks to the treatment. So if we look, for example, at the blue clone, the major clone, you can see that by day 5, when the patient received steroids only, is already decreased by half. And then at day 11, after the first dose of chemotherapy, this clone is still present, but almost completely eradicated. And by day 35, after the completion of the first cycle of 4 weeks of chemotherapy, this clone is completely eradicated. For the other minor clones, it's a similar story. These clones are also eradicated at minimal residual disease, but you can see that for one of them, this dark blue is not only not completely eradicated, but it seems that it's even expanding. So now this is worrying, and we can really detect this clone early on. So if you look at time 0, this clone was already detected at a very small frequency, while about 1% of the cells, both in blood and in the bone marrow. If you look at the mutations of this clone, it seems that it's a very different clone compared to the rest. So it's the only one that has a mutation in the zinc finger protein gene. This mutation only appears in this clone and doesn't have a mutation in Notch1 or WT1. So it seems that for this patient, there was a different pattern of evolution early on, so that the leukemic cells diverge early on into cells that acquired most probably the WT1 mutation first, because it's appearing in all the rest of the clones. And subsequently, those cells further acquire mutations in Notch1. And then other cells that diverge and acquire this mutation in the zinc finger protein that somehow is giving these leukemic cells during the treatment an advantage and are not responding. So as I said, the good thing is that Tapestri allows us to detect this clone early on. So this is an example of leukemia evolution during the treatment, but during short time. So finally, I would also like to show you an interesting case to see how the leukemia evolved over a longer period of time and how is it at the relapse. So now this is another patient from which we have 4 samples over 3 different time points. So the patient, after 2 years of treatment, relapsed. So we had the bone marrow and peripheral blood sample there. And after the patient started again with the treatment, 4 months later, relapsed again, and unfortunately passed away. If we look at the clonal composition of this patient, now you see that many more clones come into play. If you look at the mutational pattern, it seems that really this clonal architecture is more dispersed, so that the clones are less related to the others. There is less overlap in the mutations, and it seems that the evolution is less stepwise, in a less stepwise manner compared to the other patients. If we look at the evolution of these clones, we see that first of all on the left, at diagnosis in the bone marrow sample, we see a major clone in blue that has a Notch1 mutation. And this clone, once the patient started with the treatment and at the moment of relapse, it was completely eradicated. But now we see that 2 years later, the clones that govern the leukemia are a different one. So we see in red another major clone, and this one also have a Notch1 variant, different to the previous one. Interestingly, again, this major red clone that is detected at relapse, it was already detected at diagnosis in a very small proportion, only 4 cells, but it was already there. And then we also see a collection of clones appearing in this relapse sample, and again, the composition very similar in bone marrow and the relapse. Many of these clones have Notch1 mutations, and upon treatment again, all these Notch1 clones disappear. Also, we have a combined clone that has all the Notch1 signaling mutated, like the FXW7. And what we observe is that in the second relapse, now the clones that we can detect are different ones from the first relapse. We still see some residual clones from the first relapse, but now the major clone is the purple one, which has a mutation in P10. And then it seems that even P10 has further branched into another clone that has P10 plus JAK3, or other cells acquire, besides P10, also a mutation at TP53. And all these clones were not detected, nor at diagnosis and nor at the first relapse. So this is an example that over a long time, it can be that even if it's from the same patient, the leukemia at each relapse can be really different, or at least is composed by clones that seem unrelated to each other. So with this, I would like to sum up and draw some take-home messages. You have seen that ALL is heterogeneous at diagnosis. We typically find a major clone and some other minor clones. Then we observe same clones in blood and bone marrow at similar frequencies. And we have seen that different clones can have different sensitivities to the treatment. Those clones that are more evolutionary related respond similarly, but we also related clone having a mutation in the gene, in the zinc finger protein, that respond to the treatment. And I also have to mention that we have detected this same mutation in another of the patients that we are analyzing. Good news is that it seems to be feasible to detect rare but very rare clones at diagnosis, but that could be really relevant. And the leukemia we have also observed that can be completely different at relapse from diagnosis. So with this, I hope I convinced you that Tapestri can be a powerful technology to disentangle the clonal architecture of leukemia, but also apply to other cancer malignancies. And then, of course, a lot of future work to do, above all on the analysis side. We are working together to make some more complex analysis, like hierarchical clustering, where we can include more variants so that we don't lose many cell genotype information. We are also exploring different methods of variant allele mutation. We are exploring dimensionality reduction techniques to visualize all this more complex clustering. Then, of course, we want to build the phylogeny tree for each of the patients so that we can reconstruct how was the leukemia evolution prior to this. And this we want from each of the diagnostic samples from each of the patients. So that we can, for example, what mutations are more driver mutations of the leukemia, which ones occur first, and so on. Then, of course, we want to do cross-analysis. So it's very important that although the patients can be really different, try to find which clones could be common and what could be the implication of each of the clones and further study them. We have a lot of also ongoing work sequencing more patients. We also collected BLL and other samples, which I didn't show the clonal architectures today, but we are also building reference maps and diagnoses for all these groups. And then, of course, we have to integrate and include all the RNA sequencing part, which I didn't talk about today, but that data that we have for each of the samples. So with this, I would like to lab Jan for providing for all the support and providing me the opportunity to do this project, all the people collaborating with the project, Mission Bio for sponsoring this webinar, the University Hospital Leuven for the precious patient samples, the TailWatch team from VIB for the early access to this technology, all the funders of the project, and thank you all for your attention. And now I would like to give back the word to Julia.
Thank you very much, Lucia, for this very nice presentation. And let me just mention again, if you have a question, please type it into the Q&A panel. And then we would also like to ask our attendees to take a brief moment after the webinar has ended to take our exit survey and provide us with your feedback. So with that, let us join with the Q&A. And joining us for this is Hannah Biernes, who is the Senior Director of Marketing Development at Mission Bio. So let's see what we have for questions. There was a couple of questions about the tools you used, first of all, for mapping. What platform did you use for mapping? And also, what tool did you use to find the homozygosity or heterozygosity of the clones?
Mm-hmm. So maybe I can start answering that, and maybe Hannah can complete it a bit. So to map the different breeds, we use this software provided by Mission Bio. It used the BWA aligner, and this is integrated in the Bluebees high genomic platform. So it's really integrated, and we can run parallel samples there. And then for the call of the variants is used the GATK annotation method, and based on really the variant allele frequency, to distinguish the homozygous versus the heterozygous clone.
All right, Hannah, anything you want to add to that?
No, that was perfect.
Okay, great. Can you say something about the relationship between the single-cell DNA sequencing and the single-cell RNA sequencing data? And is it possible to infer the cell of origin carrying specific mutations within the different subclones?
Mm-hmm. Yes, so that's definitely ongoing work from having the clonal architecture at diagnosis. We want to try to infer the order of acquisition of these mutations, making some assumptions, like, for example, those mutations that are present in more cells will appear first, and so on, and see the relationship of the different clones. And in regards of the two data, the single-cell RNA sequencing and the DNA sequencing, we have it for both samples. Unfortunately, it's not from the same cell, so we cannot directly integrate the data. Right. But of course, they are complementary, and we can learn from each other. So the first thing we can check is like all the genes that are from which we see variants, how is the corresponding expression in the RNA sample of that patient. So, for example, this PTEN clone that we see second relapse, is PTEN expressed there, and which consequence can it have? Does the cell, this is a tumor suppressor gene, so if it's not there, maybe the PI3 kinase AKT pathway is upregulated, or what is the consequence with all the signaling pathways? Like, for example, if the zinc finger protein is lost in some cells, and which cells lose it, do they upregulate all the genes that are repressed or targeted by mRNA decay by that protein. So we can for sure make some, not directly, but some association that will be really helpful to understand the leukemia, but not only the leukemia, because we are also monitoring how the healthy hematopoiesis and the normal cells are appearing. So the RNA sequencing really tell us what are the cells and the proportion that of healthy cells and which cell types we are sequencing.
Right, okay. Quick technical question for you. What type of sequencer did you use for your experiments?
Yeah, so for all these experiments, we used the NovaSeq 6000 with the S2 flow cell, and we pulled 16 samples together.
Okay, great. Technical question for Hannah. So is this AL panel currently available, or when will it become available? And also, what's the minimum number of samples that you need to use on the Tapestri?
Yeah, great questions. So this ALL panel, you can get that today through our Tapestri designer, through our custom offering, and we are planning on making it more available as a commercial panel in the Q4 timeframe.
Okay, thank you. Question for Lucia or Jan. For unrelated mutations in the same patient, is there any evidence of germline mutations that have anything to do with DNA repair genes or similar genes?
Yeah, so far in this first analysis, we have excluded those mutations. I mean, we observe many mutations that are present in 100% of the cells, but for the clonal architecture now, we really focus on intermediate to low frequency or occurrence of the variants. Of course, if a mutation is present in all the cells, can be either germline or in the whole hematopoietic system. So it could be that it's a very early on mutation also in the leukemia. So we have to further explore what could be the consequence and which exact variants we see there.
Right. Mm-hmm. And can you say a word about validation of your panel and the reproducibility rate?
Yeah, as I said, we sequenced already 24 samples, and in terms of the performance, we always observe the same high and low performing amplicons. We sequenced them with a very similar depth, so the coverage was really sufficient. And yeah, we are able to call variants from all of the samples and all the genes all the amplicons that pass the threshold, and it worked equally well from BLL, adult, pediatric. We didn't see a lower performance for one specific subgroup of sample.
Okay. Question that just came in. What barcode libraries are currently available for research or for clinical use in other conditions? Probably a question for Hannah.
Mm-hmm. Yeah, I'm wondering, maybe they're asking about different panels that we have available. We have a couple of off-the-shelf or catalog panels that are available in hematological malignancies. So they include for AML, CLL, and for myeloid malignancies. But in addition to that, we have a custom panel offering. So if you go to Tapestri Designer, you'd be able to create your own custom panel for heme malignancies or for solid tumor profiling also.
Okay, great. And then, so how much bioinformatics knowledge does the data analysis require for these types of experiments?
Mm-hmm. So for the first part of the data analysis, there is no bioinformatic knowledge required because the Tapestri platform is really straightforward. So you can really update your FASTQ files, and normally within 50 to 60 hours. This is dependent on the number of cells and amplicons per panel, but you can run many samples in parallel. I did up to 16. But then, of course, once you get the first output and the Loom files, you can do a first exploration with the Tapestri inside, but it's really convenient to have bioinformatic knowledge if you want to get more of the data, do additional reformatting of the tables, filterings, and more complex plots or analysis. So definitely not for the first step or some of the features, but for a complete analysis, it's really required, I would say.
Right, thanks. And then finally, another quick technical question. How do you discriminate between variants that are due to amplification or sequencing errors?
Mm-hmm. Right. So if a variant is due to, for example, a sequencing error, this will be already discarded during one of the first filterings, so that we set the threshold that at least 20% of the reads need to be of the variant. So if one or two reads have a variant, is not sufficient to be annotated as a variant. So it could be a sequencing error. But if the error comes from the amplification, then all the reads of that cell will have the error, but probably it will be a very rare mutation. So it's very unlikely that two or more different cells will have exactly the same mutation at the same position. So either we expect it in a very rare proportion of cells, that then we have other samples at different time points. We don't see this mutation coming back again. Or it could also be that, that it's very infrequent and it's not annotated. That, for example, it can be a completely novel mutation where we don't have information annotated, like pathogenicity scores and so on.
Okay. Question for Lucia. Do you have plans on building similar panels that focus on the BAL subtypes?
Yeah, sorry, maybe I didn't emphasize enough, but currently this panel contains genes that are both in BLL, found in BLL leukemias and also in TLL leukemia. So it includes genes that are present in both, but if you realize also like PAX5 or EBF1 gene that is exclusive for BLL. So yeah, this panel can be definitely used for BLL leukemias, both adult and pediatric.
Okay, thanks. Here's a question that came in partial. Let's see if it makes sense to you. Can you capture interactions between mutations or other, and now something is missing, pathway zinc finger mutations?
Sorry, do you mean to link the consequence of the mutation with the pathway, or to see the biological?
Maybe that's, the question is incomplete. That's what it might mean. Mm-hmm.
Yeah, I guess that's something definitely that we have to do. So review each of the variants and see, for example, if there is a loss of this zinc finger protein, what will be the direct implications. So we expect that since this protein has an important role on the stability of mRNA, so destabilizing, the mRNA of many growth factors and tumor suppressor genes, including Notch1. A direct effect could be that if a cell loses the zinc finger protein, we see an upregulation of Notch1, and this can be another mechanism by the cell maybe doesn't need a Notch1 mutation, and without having a Notch1 mutation that makes it hyperactive for this pathway, by having a mutation in another gene that doesn't suppress it, we see more mRNA of Notch1. So for sure, we have to try to see all these indirect effects of the mutations and the variants.
Okay, thanks. And then just to clarify, your panel tests are for research use only, right? There's a question whether or not you have reimbursement, but this is not a clinical test, correct?
This is correct. Yes, that's we only use it for research, and I don't think there is any reimbursement at the moment for healthcare or by healthcare insurance. That could probably come in the future, but at the moment we use it for research.
Great. Okay, so it looks like this is all the questions we have for today. So let me thank our speakers, Jan Kuhls and Lucia Alberti-Cervera, and also Hannah Viennes, and our sponsor, Mission Bio. And as a reminder, please look out for the survey after you log out to provide us with your feedback. And finally, if you missed any part of this webinar or you would like to listen to it again, we will send you a link to an archived version via email. So with that, thank you again for attending this Genome webinar.